I will be discussing how gut microbiota impacts childhood malnutrition, as evidence suggests that there is an association between the gastrointestinal microbiome and the risk of and resilience to malnutrition in one's early life. It is important to understand the differences between malnutrition and undernutrition. Malnutrition results from specific nutrient deficiencies or diets that include inappropriate combinations or proportions of food and excess nutrient losses or utilization. Undernutrition results from inadequate dietary energy intake, which happens regardless of whether any other specific nutrient is a limiting factor. Malnutrition is a larger category that includes undernutrition. Malnutrition refers to imbalances, excess, or deficiencies in a person's energy and/or nutrient intake. The term malnutrition addresses three broad groups of conditions: undernutrition, micronutrient deficiencies, and being overweight. Undernutrition includes wasting, which refers to a low weight for a given height, stunting, which refers to a low height for age, and underweight, which refers to a low weight for age. Micronutrient-related malnutrition refers to micronutrient deficiencies, which include a lack of essential vitamins and minerals, as well as an excess of micronutrients. The last category is overweight-related malnutrition, which includes obesity and disease-related non-communicable diseases such as some cancers, heart disease, diabetes, and stroke. The primary causes of malnutrition in children are the result of inadequate access to food as well as disease such as diarrhea. Both result in deficient growth and development. Gut microbiota is also a contributing factor, which I will cover in greater detail in a few slides. Household food insecurity, poor maternal and child care practices, and poor access to healthcare and sanitation are directly related to disease and inadequate access and intake of food, while resource accessibility as well as political, cultural, and economic impacts are the root causes. Diarrhea is a major factor causing malnutrition in children, as there is a bidirectional relationship between malnutrition and diarrhea. Furthermore, alterations in gut microbiota are associated with diarrhea. Diarrhea leads to the malabsorption of essential nutrients and a reduction in available energy, which causes weight loss and can lead to malnutrition. According to the World Health Organization, United Nations Children Fund, and the World Bank Group, in 2017, 22.2% of children under the age of 5 had stunted growth, and 7.5% of children under the age of 5 were wasting globally. About half of the deaths of children under 5 years are the result of chronic malnutrition, resulting in 3 million child deaths every year. As seen on the map on this slide, the impact of malnutrition is concentrated in Sub-Saharan Africa and South Asia. Gut microbiota refers to the complex and dynamic populations of microorganisms in the human gastrointestinal tract. Altvez et al. Explains that the human body contains many microorganisms, including a large number of bacteria, viruses, fungi, and protozoa, which are referred to as the microbiota. Developing gut microbiota or microbial colonization begins at birth, which I will discuss further in the next slide. According to Arrieta et al., it continues to develop and modulate in species abundance for about 3 years until the microbiota becomes adult-like. Thursby and Young explained that the intestinal bacteria play a crucial role in maintaining immune and metabolic homeostasis and protecting against pathogens. Therefore, gut microbiota influences the host during homeostasis and disease. The diagram on this slide references the stages of microbial colonization in the infant and child intestine. The most abundant bacterial families are shown in circles, with the size of the circle being proportional to the relative abundance of the bacterial taxa at each growth stage. In newborns, the intestinal microbiota is colonized by enterobacteria. Following this stage, strict anaerobic bacteria begin to dominate the microbial community. Anaerobic bacteria are bacteria that do not live or grow when oxygen is present. In the first month, bifidobacterial species are the most predominant in the gut. At 4 to 6 months, when solid food is generally introduced, there is an expansion of clostridial species such as Lactospiraceae, Clostridaceae, and Ruminococcaceae. Clostridial species are obligate anaerobic bacteria that are normally found in our environment or the gastrointestinal tract of humans and animals. In the following months, Ruminococcaceae further develops. By ages 2 to 3, the microbiota mainly consists of Bacteroideaceae, Lactospiraceae, and Ruminococcaceae, which remain stable into adulthood. While gut microbial symbiosis positively impacts immune and metabolic homeostasis and protection against pathogens, gut microbial dysbiosis occurs when there is an imbalance of the gut's normal microbiota. A plethora of factors affect microbial colonization of the infant gut microbiota after birth. These factors include gestational age, mode of delivery, maternal body mass index and gut microbiota, infant feeding practices, antibiotic exposure, infant genetics, random exposure to microbes in the environment, and an infant's number of siblings. Infants born vaginally receive their initial gut microbiota from maternal, vaginal, and fecal sources, whereas infants born by cesarean birth receive initial colonization of the microbiota through their mother's skin. According to Drusa et al., studies show that infants born by cesarean birth have delayed microbiota colonization. Furthermore, childhood malnutrition and altered gut microbiota during childhood impact gut microbiota into adulthood, as it is linked to the development of certain diseases and disorders such as inflammatory bowel disease, mood disorders, obesity, autism, and psoriatic arthritis. Breastfeeding has a significant impact on the gut microbiota of an infant. Breast milk contains a diverse microbiome as well as oligosaccharides, which are simple sugars that have prebiotic properties. The major impact that breastfeeding has on an infant's gut microbiota comes from breastfeeding increasing the predominance of bifidobacteria in the infant's first year. Moreover, the composition of the human breast milk microbiome varies depending on the duration of the pregnancy, mode of delivery, maternal BMI, geographical location, and lactation. Furthermore, later in life, an infant and child's gut microbiota are impacted by their diet. At around 6 months of age, there's an increase in the diversity of an infant's gut microbiota related to when complementary foods are introduced. These foods are often high in protein and fiber. Differences in one's gut microbiota are associated with different post-weaning diets. This diagram represents the changes that occur in the gut microbiota that are associated with childhood malnutrition and the reductions in beneficial species and increases in harmful species. The arrows represent the factors that increase or decrease during childhood malnutrition. Evidence suggests that in dysbiosis, intestinal microbes can play a role in the predisposition of extreme nutrient status in an individual. Therefore, optimizing lifelong health can come from altering the intestinal microbiome in the first 1,000 days of an infant's life through breastfeeding, vaginal delivery, avoiding unnecessary exposure to antibiotics, and appropriate complementary feeding. Childhood malnutrition has long-term consequences, including stunted growth, lower performance in school, and impaired mental development. Reducing malnutrition in the current generation of children will be of greater benefit to the health and well-being of the next generation. It is imperative that childhood malnutrition is addressed globally. While it is important to address the root of the problem in terms of the social, economic, and political factors of malnutrition, the only clearly defined solution refers to establishing proper nutrition early in a child's life. Lancaster lays out 6 rules for nutrition that can be implemented and utilized globally. These include ensuring pregnant mothers have adequate nutrition, promoting exclusive breastfeeding for 6 months and regular breastfeeding until the age of 2, introducing mixed feeding at 6 months, continuing to feed children when they are ill, preparing, cooking, and storing food correctly, and avoiding harmful and inappropriate foods.
Right. Thank you very much. No questions in the chat so far. So we're going to let Dr. Farley try and handle the Q&A so I can get to the next presentations. So Q&A in the chat, Q&A in the room.
Does anybody in the room have a question?
I have a question for Julia. Julia,
Yes.
Now that you've done all this research about how this affects childhood, did you, when you were doing your research, see anything about how this kind of continues out into adulthood? Is this something that makes a huge difference in really an adult's life because of how childhood affects them?
Yes, well, partially just how much that the first 1,000 days has such a significant impact on how the development of the gut microbiota is impacted into adulthood and how crucial those first 1,000 days are in that lifelong impact.
Awesome. Thank you.
Thank you.
My question is, how can this be seen and diagnosed early on when it can still be treated?
That is a good question, and I don't think that I have enough information or am in the place to know how to answer that. But I think based on what I found, most of it comes from access and ensuring that people have equitable access and that impacts that, and making sure everybody has access to the opportunity of having a sufficient nutrient status.
All right. Thank you very much. One more round of applause. Woo!